Okay, so chapter 4, Eukaryotic Microbes. That's where we're starting. All right, so remember that when we were talking last time, we talked about prokaryotes, and we said that they were very simple organisms, basically a bag with everything just kind of shoved in there all together. Prokaryotes and eukaryotes are thought to evolve from the same precursor cell. So they call that cell where both of them kind of shot off of as the last common ancestor. The last common ancestor was neither prokaryotic or eukaryotic. It was kind of a combination of the two, but it did give rise to both the prokaryotes and the eukaryotes. One of the biggest held beliefs is something called endosymbiosis. And I'll explain in a bit more detail why we think this in just a second. Um, we believe that a more complex cell type most likely emerged when the last common ancestor, that cell that they both evolved from, engulfed or ate a small prokaryotic cell or cells and basically coexisted with them. A symbiotic relationship where both benefited. Now, one of the reasons that we believe that, one of the reasons that we kind of hold that as a truth is because um, mitochondria, for example, which is one of our organelles inside of our cell, mitochondria actually have their own genetic code and they have their own ribosomes, like an independent cell. Their ribosomes are also the size of the ribosomes you find in prokaryotic cells. So you've got this organelle that has its own genetic material, has its own ribosomes that are different than what you would find in a eukaryotic cell, and is actually double membraned. So with um, a mitochondria, the inside kind of looks like a squiggly, okay, which is the inner membrane. But this outside is a second membrane. So what they think happened, okay, is that you have a small prokaryotic cell, a big eukaryotic cell, and it engulfed it. It ate it. And when it ate it, this was the membrane of the prokaryotic cell, and this outer membrane was the invagination of the eukaryotic cell membrane. The genetic code also kind of lends itself to the idea that this is, excuse me, this is a prokaryotic cell originally. All right. So this is what I'm talking about. Mitochondria of the eukaryotic cells resemble a prokaryotic cell. They contain a circular chromosome, which is what you find in bacteria. They are capable of dividing by themselves without necessarily having to divide with the cell. They have their own prokaryotic ribosomes. They also have bacterial membranes that are inhibited by drugs that only affect bacteria. So the antibiotics that we have that are designed specifically to target certain bacteria actually affect the mitochondria inside of a eukaryotic cell. Now, in plants, chloroplasts are kind of the equivalent of mitochondria. They most likely arose from that same type of symbiotic relationship. Probably a cyanobacteria, which is one of the microbes that use light to make energy or to make food. So the cyanobacteria provided their host cells basically with a built-in feeding mechanism. Mitochondria are fantastic at making ATP, which for our cells is our fuel source. It's like saying gasoline. So having something that makes ATP in huge amounts is very beneficial to our cell. In fact, out of all of the ways that we make ATP, the mitochondria are the most efficient and they are also the best at it. The best way I can think to describe this, we basically have three primary ways in our body that we make ATP. The first, you get one ATP. That's like saying you get a quarter. The second, you have glucose as a fuel source, and when you break it down, you get 2 ATP. So you get 2 quarters, okay? In a mitochondria, one glucose, one sugar, will give you 36 to 38 ATP. So 36 to 38 ATP is quite a bit more as far as the payoff. That's 36 to 38 quarters versus 2 or 1. Which one would you rather have? Of course, you'd want to have the 36 because it's more money, right? Having something inside of you that makes the ATP like that you can use is fantastic. Same with, excuse me, the chloroplasts. The chloroplasts have the ability to use light to make sugar, basically. So having that ability to do that is very beneficial for the bigger cell because they can take advantage of the fact that they're making food that I can actually eat. All right. So, eukaryotic microbes. Okay. The main pathogenic, remember pathogenic means that it'll make you sick. Pathogenic eukaryotic microbes we will be discussing are going to be the protozoans, the fungi, and the helminths. Okay? So, the structures inside of our eukaryotic cell. Note which structures are always present. Note which structures are present only in some of the eukaryotic cells. So every eukaryotic cell is going to have a cell membrane, a nucleus, mitochondria, endoplasmic reticulum, a Golgi apparatus, a cytoskeleton, and lysosomes. Lysosomes, lyse means to break. These are little bubbles that have digestive enzymes in them. When things break down in the cell, you kind of shove it in there, it gets digested, okay? The nucleus with a nucleolus inside, which is a dense little body inside that you will see in just a bit, and a nuclear membrane. In other words, there is a wall, a membrane, surrounding where the genetic material is. It's isolated from the rest of the cell. Remember that in prokaryotic cells, that didn't happen. Literally in prokaryotic cells, you just have it kind of floating around. There's kind of a general area that it sticks to, but it's not like it's encased in its own space. Okay? The endoplasmic reticulum has two types, rough and smooth, which we will talk about, and the cytoskeleton. Now remember that we talked about the cytoskeleton as something that kind of helps the cell hold its shape. Our skeleton helps us hold our shape. Cytoskeleton helps the cell hold its shape. With the cytoskeleton of a eukaryotic cell, you've got actin filaments, you've got microtubules, and you've got intermediate filaments, each of which is slightly different from the other. Now, what are present in some of the eukaryotic cells? Cell walls. Cell walls are going to be more common in our prokaryotic cells, okay? But some eukaryotic cells do have them. A glycocalyx. We kind of talked about this as kind of an outer layer that has sugars on it. Centrioles. Centrioles are used for dividing your cell, which we'll talk about. The chloroplasts. Chloroplasts are going to be in things that use light. We have no chloroplasts. We are not green. We are not a plant. Okay? And then flagellum and cilia. These are present in some eukaryotes, but not in others. We don't necessarily have flagella anywhere except the sperm. That's like the one exception. Cilia we have, but not for the same purpose as a single-celled eukaryotic organism. Okay, so if you want to see where everything kind of fits, This is a good flow chart to look at, okay? It's a great way to kind of look at what's what from outside to inside. Try to organize the structures like this in your mind. Also, be sure to note that the slot in the slide you can see, you can mark what structures are optional. Remember, I said in some, but not in all. Okay. So let's talk external. So we're working our way from outside to inside. Appendages for moving your cell from point A to point B. The flagella. Now, compared to a eukaryotic cell, compared to a prokaryotic cell, the flagella in a eukaryotic cell is more complex. It's thicker as well, and it's made out of microtubules in a 9 to 2 plus 2 arrangement. So microtubules are kind of like a hollow pipe, okay? And you can actually see that here. 1, 2, 3, 4, 5, 6, 7, 8, 9. And then two hollow pipes in the middle. Okay, 9 plus 2 arrangement. You can see it here, and then there's the two pipes. Okay, so it's a bit more complex, which makes sense because eukaryotic cells are more complex cells. Cilia. Cilia are not long whip-like tails. Instead, you've got these short little hairs, almost like looking like velvet. Okay? They're shorter, they're more numerous than flagella, and they're mainly for movement, but can be used for feeding. So these actually kind of create a synchronous movement where they all kind of move together to get the cell to go where it wants to go. It can also act as a way to get it to and then kind of capture food. Okay? Compared to the flagella, these are on more surface and they have to work as a group instead of one big propeller that moves you. Okay? Now, I talked about the glycocalyx being optional. It is found in most eukaryotic cells, but because it's not found in all of them, we don't include it in that umbrella. The outermost layer of your eukaryotic cell, this is the outermost layer of your eukaryotic cell, and it is in direct contact with the environment that this cell is sitting in. So if we're talking about the outermost crust, that's going to be the glycocalyx. It can also be called the extracellular matrix. So, what does it do? Why do we have it? Well, for one, it protects the cells. Okay? So, it's an outer kind of shielding for them. It can help adhere the cell to surfaces. Remember, I've got those sugars sticking out. It can actually help anchor this type of cell to a surface. Lastly, it can help with cell to cell signaling, signaling. So each glycocalyx, each covering is unique, kind of like a fingerprint. So being able to identify something as matching you, it's the same as me, or being different from you is a way to signal between cells. Okay. So it is a way for cells to identify other cells as being the same or being different. All right. So the boundary of the cell. Our cell wall versus our cytoplasmic membrane. There is a difference between these two things. So you'll hear cell wall and cell membrane, two completely different things. It's like saying a motorcycle versus a pickup truck, completely different things, okay? So for our cell wall, not all eukaryotic microbes will have this. As I said previously, in our list of what every cell has and what some cells have, cell wall isn't in everybody. Fungi are the only ones that really do have a cell wall. Protozoans and helminths do not. Okay. So in the fungi, the cell walls are rigid. Remember, I said the cell wall is like bricks, it's not flowy or anything like that. It's actually pretty solid. It is used to support the fungi cells. Okay. And they're actually a lot different from what we see in the bacteria. Remember that in the bacteria, we've got the proteoglycans and we've got gram positive and gram negative. These are actually way different. They have this thick inner layer of something called chitin. They can also use cellulose, okay, with a very thin outer layer of mixed glycans or polysaccharides. So you've got kind of a mixture of things instead of it's just proteoglycans, the end. Instead, you've got the chitin or cellulose and you've got glycans as well as a mixture. So when you look at a fungal cell, here's the membrane, so the inside. You've got chitin, glycoproteins, glycans, and the glycocalyx. This is not proteoglycans, okay? So the recipe for the cell wall in the case of fungi is very, very different. Now, our cytoplasmic membrane. This is very similar to the prokaryotic cytoplasmic membrane. You have a bilayer of phospholipids. Now remember, phospholipids are the ones that have the head with the two tails that are kind of huddled together in the middle. I talked about it being kind of like a basketball, okay? Where if I touch the outside of the basketball, I'm touching heads. If I touch the inside of the basketball, I'm touching heads. But that layer in between, that outside and the inside, would be where the tails are. So it is a phospholipid bilayer, same makeup. We do have embedded proteins inside, and it serves as a selectively permeable barrier. Selectively permeable. I select who gets to go. I don't let things go that I don't choose. Okay, it's selectively permeable. How is it different from the prokaryotic cytoplasmic membrane? Um, it contains sterols that gives the cells stability. It's helpful for the cells that don't have a cell wall. So remember I mentioned sterols. They actually kind of help so they don't, it's not so unwieldy, lack of a better way to say it. Okay, remember I talked about a swimming pool with balloons on it. You hit the water, the balloons move all around, and that's kind of the way that I think of a cell membrane. When you add cholesterol or sterols to that, it's almost like adding big pieces of duct tape that are holding sets of balloons together. So instead of moving as independent balloons, you kind of have islands of balloons moving when you hit the water. It gives a certain amount of stability. Now, does it make it hard like a shell? No. But it does give it a bit more stability so that instead of independent phospholipids, you've got kind of islands of phospholipids that move around. The sterols are going to help that to happen. In our cells as humans, we actually have cholesterol molecules that do this for us, kind of help to stabilize the surface of our cells.